Hello, everyone, and welcome to today's presentation. This webinar will have two parts. Part one is Advanced Gene Therapy Development Solutions, a comprehensive platform for next-generation therapeutic innovation, an overview of GenScript's gene therapy solution. And part two will be a deep dive into GenScript's viral vector solutions for delivery of gene therapies, titled Novel Viral Vector Solutions. This webinar is a part of the second annual Cell and Gene Therapy virtual event series. My name is Delia Osborne with LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with others in the live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of our presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within this event. Now, please, let's get started. I'd like to present our first speaker, Roumen Bogoev, followed by Dr. Xiang. For a complete biography on our speakers, please visit the Presenter tab from the menu on the left of your screen. Roumen, you may now begin your presentation.
Thank you, Delia, for the introduction. It is my pleasure today to introduce our attendees to the GenScript Advanced Gene Therapy Development Solutions. In the first, I'll start with the agenda table of contents. I'll introduce briefly GenScript as a company, if you're not familiar with the company, then speak about the overview of the gene therapy solutions that we offer, then focus on the gene therapy portfolio. And my introduction will be more overall introduction of the solutions that we offer and focusing on the non-viral gene therapy solutions mostly. And then one of my colleagues will introduce you to the viral solutions that we offer for ex vivo gene therapy. So a little bit about GenScript. GenScript is a global company. We operate in many regions of the world. We operate in over 100 countries. We have over 200,000 customers around the globe. We have more than 6,000 employees and 800 R&D staff. We have many patents and patent applications. And we basically service many customers that have published using our services and products. And we have over 100,000 citations in prestigious journals. We have many manufacturing sites around the world. We have 2 manufacturing sites in the United States, 1 manufacturing site in New Jersey, another manufacturing site in Seattle, Washington. We have multiple manufacturing sites in China. We have manufacturing site in Singapore, serving our Asian Pacific customers. And we have many commercial offices around the world. We have, of course, commercial offices in the United States, United Kingdom, Netherlands, Korea, Japan, Singapore, and other countries around the world. So the GenScript group consists of several entities. Today, I'm not going to speak about all these entities. My talk will be focused on the entities on the left side, focusing on GenScript Life Science Group and ProBio Biologics CDMO division. So very briefly to introduce the entities on the right side, Bezyme is our synthetic biology division. They have very strong synthetic biology capabilities and focused on human and animal food supply. Legend Biotech became public a few years ago on the New York Stock Exchange. They're an innovative cell therapy company. They developed Carvykti together with Janssen that has been launched a few years ago. And now they have advanced pipeline of solutions for leukemia, solid tumors, infectious disease, and so on. So My focus will be on the entities on the left side, the life science group, which consists of our CRO division as well as the product division. They offer a lot of innovative services and products for therapeutic developments. We use a lot of automation internally in our CRO operations to boost our efficiency. We also sell a lot of automation solutions to our customers in form of advanced instrumentation, as well as we offer many reagents to go with this instrumentation. In our biologics, ProBiobiologics CDMO division, they have very large-scale GMP capacity to meet the market demands. They have very strong regulatory experience working with many customers, starting from R&D all the way through clinical trials and commercial manufacturing. They have CDMO solutions for protein drugs, antibody drugs, plasmid drugs, mRNA, viral solutions, and others. In addition to gene therapy development solutions, we also offer solutions for many other therapeutics like cell therapy, antibody therapeutics, vaccines, and IVD. Please watch our webinar also by Daksha Patel on cell therapy development solutions if you're working in that area. So quick overview on gene therapy solutions. As we all know, most therapeutic developments start with sample analysis, basically discovery and validation of the target genes. Many patient samples are analyzed in different ways, in vitro and in vivo, and we offer a lot of solutions for that part of the development workflow. And then regardless how you are going to proceed with ex vivo gene therapy, in vivo viral gene therapy, or in vivo non-viral gene therapy, we have solutions for all of those that we can offer. So this is a very quick overview of our integrated solution for gene therapy development. We can offer a number of solutions for target discovery, for target gene validation, for viral lead generation and optimization, for non-viral lead generation optimization, for preclinical studies, for R&D support, as well as clinical trials and GMP manufacturing. So I'll go in a little bit more detail of some of the key services and products that we offer for gene therapy development, starting with our gene synthesis service. Of course, gene synthesis is a fundamental service that is applicable to many therapeutic developments. We have been offering gene synthesis starting since 2008. We have hundreds of thousands of customers for this service. We have delivered over 3 million genes so far with 99.95% success rate. And we have very large capacities to synthesize genes. We can synthesize over 200 million genes per month, 200 million bases per month. And we have global production network, as I mentioned, with multiple manufacturing sites in different regions in the world. In addition to the gene synthesis, we offer also free-to-use GenSmart codon optimization software. This software uses a number of parameters to optimize your sequence for expression. We also have GenBrick gene synthesis service, which is for very large DNA fragments, where we can synthesize the very large fragment into multiple fragments, and then you can seamlessly integrate it with our cloning kit. In addition to the synthesis services, we also offer, for our customer convenience, we offer VectorArk and CloneArk storage systems. Those allow you to store your vectors or your clones with us for easy reuse for future projects. Our GenSmart codon optimization software offers very advanced methodology for analyzing your sequence and optimizing it. It's free to use for our customers. It's integrated in our ordering system. It uses over 200 factors that are pre-screened to optimize your sequence for optimal expression. And of course, it goes together and can be used together with our VectorArk and CloneArk storage systems. And we have many happy customers around the world, including Dr. Liu from Harvard University, who is very happy with using our gene synthesis and codon optimization software services. Here is a quick case study about our gene synthesis and codon optimization. Here we are showing how powerful codon optimization can be for increasing your expression capabilities. We compared 3 clones from each GFP and JNK3 proteins with wild type and also codon optimized. And you can see on the top here, you can see how much stronger is the expression of JNK3 with the codon optimization, also of the GFP with the codon optimization compared to the wild type. So codon optimization can be a very powerful tool for you to increase your expression. GenTitan technology is one of our technologies we use for oligosynthesis. Is very capable to synthesize very large oligo pools. We use CMOS chips, and we can synthesize basically either 92,000 or 8 million oligos at the same time in a single chip. And we can use this technology to generate a lot of libraries. We can use them for NGS enrichment libraries, hybridization-based enrichment libraries, amplicon-based enrichment libraries. We can also use it for our CRISPR gene RNA libraries. We also develop a lot of mutant libraries for lead optimization. We have, using our GenTitan technology, we can develop site saturation libraries, saturation scanning libraries, combinatorial libraries. Basically, we start with the GenTitan synthesizing the very large number of oligos, turning them into a plasmid, amplifying, and then either offering it as a plasmid library or as a viral vector library in the end. This is a very quick study where customer approached us with the need to create NNK library for optimization of AAV5. They wanted to increase the tropism of AAV5 into the liver. We created the library for them, and they were successful in finding a clone that actually showed them much better tropism concentration into the liver compared to the wild type, and very basically with very little concentration of the virus into other tissues like brain, kidneys, other muscle tissues, and other tissues. We also offer cell line services. We have a wide variety of them: customized CRISPR knockout cell line service, whole gene CRISPR deletion service, customized CRISPR knock-in cell line service, customized CRISPR screening and off-target service, cell power gene constitutive expression, cell power gene inducible expression. In addition to this variety of services, we have also very large library of over 250 different cell lines that are off the shelf. Our off-the-shelf cell lines group in several categories. We have very large number of cell lines focused on GPCR. We also have large number of cell lines focused on FC receptors. And we have immune checkpoint cell lines for cancer research, as well as we have some cell lines for COVID research for ACE2 and spike protein cell lines. All these cell lines are single clone. They can be offered in multiple species. They have guaranteed performance, and we have very stringent QC for these cell lines. And they can be shipped to you within 1 to 2 weeks versus the 4 or 5 months that it takes to develop stable cell line. We also have very strong capabilities in vector construction. We have our GenSmart vector design software. It includes, it has basically over 200 very commonly commercially available vectors that are already built into the software, over 2,000 functional parts that are typically used for vector design. It has automatic sequence annotation, free plasmid mapping. It does error checking. Also, it's integrated with our GenSmart codon optimization software. So while designing your vector, you can also codon optimize your gene. We basically are very efficient in constructing vectors, a variety of them with very high throughput and very fast speed. This case study for vector construction, a customer contacted us. They wanted to improve AAV vector egress factor. And we basically created several clones with them with different serotypes of the MAP gene from different serotype AAVs, 1, 2, 5, 8, and 9. And after testing these different additions of MAP gene to the vector, they were able to increase the viral egress successfully. We also offer a variety of DNA preparation services, starting from research-grade plasmid preparation service, industrial-grade plasmid preparation service, preclinical-grade plasmid preparation service with different increasing the quality and the QC standards. Also, our ProBio CDMO offers GMP-grade plasmid production in our facility in Hopewell, New Jersey, as well as the facilities in China. In addition to the services, we also offer instrumentation. We have fully automated instruments for mini prep and maxi prep plasmid preparation. The mini prep instrument can purify 48 plasmid samples at the same time with a single module. Up to 4 modules can be attached to the controller system, giving you throughput of 192 samples. And basically, it has very flexible starting volume from 1 milliliter to 5 milliliters. And if that is insufficient for your research, then we also offer maxi prep, fully automated system. This system is capable of purifying up to 24 samples at a time with up to 4 modules. Each module that is attached to the controller here, each module can purify 6 samples. And the starting volume can be from 50 milliliters to 200 milliliters, 0.3 grams to 1.5 grams bacterial pellet. And both systems can provide essentially good way for you to start with automated system and slowly scale it up as your demand increases. So you can keep adding more modules, up to 4 modules for each system to increase the capacity of your system. And they both offer transfection-grade plasmid. We also have very strong portfolio for CRISPR gene editing. We offer a variety of enzymes, Cas9 enzyme. For quick refresher, of course, Cas9 does blunt and break, cleaves 3 bases away from the PAM region. After cleavage, the enzyme is released and usually prefers G-rich PAM regions, PAM sequences. We also offer Cas12a for gene editing. This one can do staggered break for better knock-in insertion, cleaves 18 to 23 bases downstream, stays bound after cleavage, and prefers T-rich PAM sequences. We also offer Cas13a enzyme for RNA editing, single-stranded break, cleaves downstream, stays bound after the cleavage. So we offer a variety of enzymes, including we are offering some of the newer modalities that are more recently developed. So in addition to the conventional Cas9 cleavage on the left side, we also offer base editing and prime editing enzymes. The base editing enzyme is characteristic with the fact that it's capable of modifying single bases very precisely and very efficiently. And many of the genetic conditions are actually caused by single base mutations. Over 50% of the genetic conditions are caused by single base mutation. And we also offer the prime editing from Broad Institute, under license from Broad Institute, several versions of their prime editors, which are also very efficient in modifying the DNA, very specific, and they use dead Cas9 bound with reverse transcriptase and can do small insertions or deletions. Some of the challenges with these novel modalities are that they tend to be relatively large enzymes, so they're a little bit harder to transfect. AAV cannot be packaged in AAV because the genome is too large. So you need to optimize a little bit more your transfection efficiency and methodologies. However, they're very precise and very efficient at the modifications. And this is basically, we have very large selection of variety of different options for Cas9, SP-Cas9, Ultra, ESP-Cas9, SP-Cas9 with GFP. We have also variety of Cas12s. We have with smaller molecular weight. We have SA-Cas9 also with smaller molecular weight to fit into AAV vector. We also have two versions of RNA cleavage enzyme, the Cas13A. We also have multiple versions of our prime editors, again, licensed from Broad Institute, P6C, P6D, P-Max, P2 prime editors. We also have base editor that converts C to T and A to G. We also have very strong expertise in custom RNA enzyme development and production. So if you need anything else that we are not offering at the moment off the shelf, we have done over 70 different versions of different gene editing enzymes, and we can very efficiently develop them and produce them for you. In addition to the enzymes, we also offer the guide RNAs. We offer guide RNAs for most of the enzymes out there, including Cas9, sgRNA, prime editing pegRNA, base editing gRNA, Cas12a crRNA, as well as we can synthesize any guide RNA that you may need, up to 266 nucleotides. In addition to the enzyme version of the Cas enzymes, we also offer plasmids. We offer single vector system that can allow you to synthesize the gRNA in Cas9 at the same time with a single plasmid, or we have dual vector systems. One of the plasmid would allow you to express the Cas enzyme, the other one the gRNA that is needed for it. And many of those have been licensed from Broad Institute. They have been pre-validated by the Broad Institute and tested for efficiency. And we have a variety of different options for them. We also offer HDR templates for knock-ins. We have 3 different versions. We offer GeneXact single-stranded DNA. This is very efficient. It has minimal cytotoxicity, is very precise knock-in in minimal off-target effects, but is somewhat limited to smaller fragments. Up to 5 bases fragments can be inserted using this single-stranded DNA HDR template. We also offer double-stranded DNA with covalently linked ends. The advantage is the covalently linked ends stabilize the template, make it more stable for scaling up and for accuracy of the modification. And also this template allows you to insert genes up to 10 bases. We also offer GenCircle double-stranded DNA. Circles are usually very efficient for knock-ins. It increases knock-in efficiency up to 30%. Has very low cytotoxicity, immunogenicity, does not have any antibiotic-resistant genes into the backbone. The backbone is only 428 bases and is very small. The circle template allows you to insert genes as large as 50 bases. So we offer a number of these in research use as well as for GMP, in GMP production. We have very efficient software for designing sgRNAs and HDR templates. In the top table, you can see we compare the GenScript software with several popular vendors on the market and showing the predicted efficiency, specificity, and off-target effects. Also here on this chart, we offer the indel ratio for each one. And you can see that overall, the GenScript templates has better predicted target rate, has higher target specificity, and at least one of the gRNAs that was predicted works very, very well with very high efficiency. For CRISPR, we offer solutions from early research to preclinical development through IND filing and clinical trials in GMP. With the corresponding quality level services and products. We have many products here for research use, then for our GMP-like services and products for your preclinical development and IND filings. And then we have the GMP hRNAs, GMP enzymes, and GMP HDR templates that we offer for clinical trials as well as commercial production under GMP conditions. And here is just one example, us supporting a client for their human clinical trial. We synthesized 2-component hgRNA for their CAR-T therapy that integrates PD-1, and they were successful with their clinical trial, getting good results, treating the patients. A lot of scientists prefer to use mRNA format for their CRISPR gene editing to have the enzyme in mRNA format for its transient nature, because the enzyme doesn't linger into the cells to do some unintended cleavages. And we offer a complete solution from beginning to end for mRNA synthesis. We can start with codon optimization. We have codon ready-to-use UTR designs that we can offer, or we can use the customer UTR regions that they prefer. We can do the gene synthesis from small to very large genes very fast and efficiently within a week. And then we can prepare the plasmid. We can linearize it to prepare the templates for the IVT reaction. We can produce any enzymes if needed that are custom, that need to be custom produced for the development. And then we can produce mRNA, either a linear mRNA, saRNA, or circular RNA. In addition, we can also package the mRNA into LNPs. And I'll show more detail about our services later. So as I mentioned, we can offer either linear mRNA format, self-amplifying mRNA format, or circular RNA, and they have different advantages and disadvantages, but typically the saRNA and the circular RNA offer much stronger protein expression. They're much more stable, have longer half-life. They have low immunogenicity, and typically they require smaller dosage. And they're very novel that we have recently introduced, and we're one of the first vendors to offer these to our customers. In addition to the services, we also have some off-the-shelf products that you can use for your testing. For example, we have a number of reporter genes in linear mRNA format, in circular RNA or saRNA. We have also gene editing enzymes already off the shelf in mRNA format, in linear and some linear format. For a number of them, ESP-Cas9, SA-Cas9, Cas12. We have some of the newer editors, prime and base editors in linear format, and other gene editing enzymes. In addition, we have some antigens in gene replacement mRNAs here in this section, and we also have WADRs and standards that you can use for your developments. We have very strong LNP services offering. We have very fast and efficient LNP services that can start from the selecting the enzyme, selecting the guide RNA, packaging them, basically producing them as mRNA, packaging them together into the LNP, and delivering with very fast, very short timelines. In addition, we offer targeted LNP services. Some of them are already off-the-shelf targets that we have introduced, like CD13, CD8, CD34, some others that are focusing on specific cell types. And we can also develop custom LNP services, custom targeted LNPs for your developments. Here is some feedback from customers. Very happy customers with our EGFP expression in cells, or mCherry expression also in cells, with the FLuc mRNA into mice, and also some customers using our mRNAs as antigens for animal immunization or customized mRNA expression. Our CDMO division has a lot of regulatory expertise, and they have done many developments in the GCT area already. They have developed over 100 batches of GMP products. They have worked with customers on over 20 successful audits in different regions around the world: China, US, Europe. They have done over 30 IND projects, and they have successfully supported 6 IND approved filings. And this is just a summary of the services and products that we offer. Starting with our antibody development, we offer it all the way from early research through scaling up, preclinical, and then IND and clinical stage. CRISPR, gRNAs and HDR templates from research to preclinical and clinical grade, plasmid, IVT, mRNA, AAV, and lentivirus production, complete from our research to GMP. Peptide synthesis also from research to GMP. We also have some instrumentation that we offer and that supports research use as well as GMP use. For example, our cell isolation system, we have some manual magnets as well as fully automated systems that can be used for research use. And there we also have the automated system as GMP grade to be used in GMP production. We also have them together with our non-toxic nanomagnetic beads for isolating different cell types like T cells, NK cells, stem cells, and other cell types. We also offer very good T cell activation reagent for research use and GMP use. And we also have as I mentioned, the plasmid purification systems that we offer for mini prep and maxi prep for research use, as well as we have magnetic bead-based protein purification system that is very efficient and can purify a very large number of antibody and tagged protein samples very quickly. And this is my part of the presentation for today, and thank you for your attention, and I'll let my colleague continue with our focus on viral developments. Thank you for your attention.
Hello, everyone. Thank you for your joining us today. Many thanks for LabRoads for hosting this valuable event. My name is Xiang. I'm the field application scientist lead at GenScript Life Science in North America, where I specialize in supporting GenScript's viral vector service. Today, we will explore the key innovations shaping the next generation viral vectors. The presentation is divided into 3 parts. First, we will explore the cutting-edge innovation that are defining the next generation of viral vector. Then I will see how these advanced vectors are being applied, illustrated with some compelling real-world examples. And finally, I will show cases and how these innovations are being integrated into the latest upgrade GenScript viral vector solutions. And I'm happy. I hope my talk is comprehensive and valuable where the field is heading. Today, the field of CAR T cell therapy is advanced in 2 distinct fronts. The first is a well-established in vivo, ex vivo method. This is an autologous process, meaning it will use the patient's own cells by collecting their T cells activation, engineering them into the lab. Mostly, we will use a lentivirus vector to express chimeric antigen receptor known as CAR. After the patient undergo lymphodepletion chemotherapy, then refuse the engineered cells back to their body. The major advantages here is this is an approved technology. Its efficacy underscored by the fact that 7 ex vivo CAR T therapy are already approved on the market. However, the process is also incredibly resource intensive. It's costly, time consuming, and it's personalized. It presents significant challenges for scalable and broad timely access. This brings us to the second emerging strategy, in vivo CAR T generation. This is an off-the-shelf gene therapy instead of engineering cells in the lab. The viral vector or lipid nanoparticles are directly administered to the patient. These vehicles are designed to target T cells within the body and deliver the CAR. The potential benefits are a dramatically faster treatment of timeline, significantly lower cost, and a process that may be more biocompatible, potentially reducing the risk of immune rejection. The challenges, however, is substantial. Such hurdle in specific targeting and transduction efficiency and the potential need for repeat dosing. So the field is needing a precise and highly efficient and safe therapy. So benefiting from the novel lentiviral vector, the journey from ex vivo to in vivo is the next great frontier, making this powerful CAR T therapy more accessible to patients. We will discussing the two workhorse of the main modern gene therapy, adeno-associated virus or AAV, and the lentivirus vector or LVV. These are most commonly used engineering viral vectors for delivering genetic material to the cells, but they have very distinct properties and applications. AAV is a non-enveloped, and its target mostly post-metatic cells or non-dividing cells, such as neural, cardiac, muscles, and retinal cells. After delivering the genetic cargo, the AAV genome typically persists in the nucleus as a free-floating circle known as episome. Because it doesn't integrate into the host DNA, it avoids the risk of disrupting important genes. This allows for relatively long-term transient expression, making a leading platform for direct in vivo gene therapy, where the vector injects directly to the patient. However, the main limitation of AAV is its small size. It only carry a payload about 4.5 kb. In contrast, lentivirus or LVV is an enveloped viral vector with broad tropism, allow it to infect both dividing and non-dividing cells. Its key feature is that a stable integration in the transgene into the host genome, which ensure long-term expression. A significant advantage of modifying dividing cells population. With a larger payload capacity than AAV, lentivirus is extensively used ex vivo gene therapy, and it's a fundamental tool for generation of a stable cell line and the functional genomics screening. However, it carries a risk of insertional mutagenesis in the genome, limiting it as in vivo gene therapy application. Moving forward, I'll walk through the key innovations shaping the next generation of lentivirus vector as an example. Let's start from fundamentals. The original lentiviral genome is a 10 kb single-strand RNA molecule packaged within a lipid shell about 100 nanometer in diameter. The scientists have utilized this a set of plasmid to produce recombinant vectors through several generations in more than third generation packaging system, as shown in Figure C. It has three main components: the transfer plasmid, packaging plasmid, and lastly, VSVG glycoprotein in the envelope plasmid that provides the viral with a broad tropism and cell entry. This field is focused, but the story doesn't end. This field is focused on next generation lentiviral vector engineering to overcome historical limitation. The driving force behind this innovation is urgent technical need for in vivo application, such as the off-the-shelf CAR therapy we discussed. A prime example of early innovation to meet these needs is pseudotyping by construction vectors with different envelope genes like VSVG or now even the banders. Scientists are creating a distinct pseudotypes that exhibit outer or more specific cellular tropism, a critical step towards generation in vivo delivery. The advantage focuses on four crucial areas: enhancing safety, targeted delivery, expanding cargo capacity, streamline manufacturing, and even more. And now let's look at how this science is translation into real-world therapeutics. The industry is pushing this concept forward in incredible, exciting way. Consider the challenge in vivo delivery that we have discussion. While many are trying to resolve this by lipid nanoparticle or standard lentivirus, this often face a critical problem of target uptake. This limits their use and efficacy. A platform called INT2104 from Interius, the scientists offer a specific cell targeting in vivo solution to the right cell and right disease through a right payload. It's an engineered lentiviral vector with three unique features. It uses a target binder for CD7, a marker T cells and NK cells. It couples with a modified VSVG, the targeted LDL receptor fusion that retains the fusion function. And finally, it includes a lentiviral transfer gene CD20 CAR designed for B cells malignancies. The data is compelling. Single intravenous dose of INT2104 can generate a powerful arm of both CAR T and CAR NT cells inside the body, capable of effectively depleting targeted B cells. And now let's look at how this science is translating into real-world therapeutics. The industry is pushing this concept forward in incredible, exciting way. Consider the challenges in vivo delivery that we have a discussion. Well, many are trying to resolve this in lipid nanoparticles or standard lentivirus. This is often facing a critical problem of target uptake. This limits their use of and efficacy. A platform called INT2104 from Interior scientists offer a solution-specific cell targeting in vivo therapy. This solution is using a right cell, right disease to treat right disease through a right payload. It's an engineering lentivirus vector with 3 unique features. First, it uses a target binder for CD7, a marker on T cells and NK cells. It coupled with a modified VSVG, the target LDL receptor fusion gene that retains the fusion function. And finally, it includes a lentiviral transfer gene CD20 CAR designed for B-cell malignancies. The data is compelling. A single intravenous dose of INT2104 can generate a very powerful arm of both CAR T and NK cells inside the body, capable effectively depleting targeted B cells. The beauty of this therapy is no need to isolate T cell from patient. This is a completely eliminate the ex vivo process and associated challenges. It demonstrates the potential truly viable off-the-shelf CAR therapy that could bypass the complete complex, costly, time-consuming ex vivo manufacturing and process. The third advantage is that patient does not need to go through the toxic lymphodepletion. Moving forward to reality, it's common assumption that generation high quality virus in-house is straightforward process. Oftentimes rely on a kit protocol. However, reality is many labs facing the challenges. Listening here, lack of the quality control, low titer of lentivirus, and the capability of a scale up and time consuming. So we just learned that about the common pain point of time and title, the consistency and the toxicity at the GenScript. We are engineering one in all lentiviral packaging platform from the ground to directly resolve this problem for you. It starts with a commitment to speed and reliability. This is just not a isn't just a claim. It's a result of a deep optimization, fully integrated system to consistently achieve high titer and quality. We guarantee first 10 to the power of 8 TU per mile functional titer. We use a proprietary innovation packaging system for large gene insertions over 10 kb, maximize 400 titer of boost. And the rigorous quality control, we use a duplex PCR to quantify measure the genomic integration for confidently give you a quality control for each batch. A critical challenge in the field of lentivirus production is obtain accurate measurement of a functional titer. Traditionally, the field usually rely on the P24 ELISA IC. This method is quantify the capsid protein, giving you a physical title account of all viral particles, both functional and non-functional. The problem, P24 significantly overestimated the true infectious title because it includes defective non-infectious particles that never transduct the cell. That's why, to provide you true accurate assessment infectivity, we have made a duplicate PCR as standard. This method specifically quantify only the virus genome that have successfully integrated to the target cell. In essence, it directly measures the infectious functional particle that capable of transduction. So by including this duplicate PCR data in each batch, we can ensure you receive a constant high quality product, give you a greater confidence in the experimental and result. As we have learned that GenScript entire pipeline and its refined quality and speed through an upgraded all-in-one packaging system for superior efficiency. Now let us share you a comparison of functional title of some commercial system versus the GenScript proprietary system. As you can see in the chart on the left, our upgrade system shown in the blue bar constantly produce a significant higher functional title compared with the standard commercial system shown in green. Across a range of vectors, you can see the constant and substantial boost in functional title. Let's put some number on it. Vector 1 more than double of title, and vector 2 493 increase, near fivefold of higher, and for vector 5 an extraordinary 567 improvement. This performance is especially critical when work with a large gene as interest. As you see, data in the right demonstrate our platform efficiently boosts the functional title even for these challenging constructs, and deliver dramatically improvement. And in short, we have turned a complex and predictable process in a reliable, rapid, and high-performance service. Now let's have a real-world example in GenScript, how GenScript resolves the challenge of oversized insertion of a lentivirus vector. The problem here is very clear. The client have insertion over 10 kb. The packaging efficiency collapse, title drops, and downstream performances suffers. So with our proprietary vector, and as showing an example here, we optimize with every step used within a control. Then the result is compelling. And just within 48 hours transduction, you can see the strong fluorescence, clear evidence with gene expression. To show you the case, our in-house capability, I'd like to share some data and customer experience how prove these performances. We have successfully handled large-scale sample in efficient and precise way. For instance, we complete an order of over 250 samples once. Each require a title of 10 to the power of 8 for 100 microliter per sample. We finished just within 10 business days. The gene synthesis is about 4.8 business days, and lentivirus package is 4.5 business days. All the sample passed the QC check and verified by qPCR and effects analysis. Meet the physical title of achieving the functional title exceeding 10 to the power of 8. At last, I will switch my gear to some key AAV engineering. Similarly, the AAV field is aiming to maximize efficacy and mitigating the toxicity in following 3 areas: the gene insertion, codonization, vector optimization and construction, including the target specific promoter and enhancing and transgene expression, regulatory elements, etc. Third, capsid engineering enables specific tissue tropism. At GenScript, we are the leader within the field, where we are expertise on one-stop shop solution. We provide you comprehensive solution on codonization, oligo pool, and mutant library service. The supporting case study on AAV codonization come from researchers citing GenScript. They were aiming to develop a second-generation clinical AAV vector. In this study, they demonstrate the combination effect, codonization and vector optimization, which significantly improved the efficacy of an AAV vector in LDL receptor a deficient mice model. In conclusion, the codonization and vector optimization enhance the transgene expression and reduce the serum LDL level efficiently. I would like to highlight the innovation here, GenScript codonization tool, in terms of CAI, GC content frequency, and codon optimization expression can be improved, leading a more effective reduction of serum LDLR level. Next, come to a future direction and summary. Ongoing development of a novel lentiviral vector aiming to make the gene therapy more accessible, effective by lowering the costs, minimize the potential side effects, improving precise control of gene expression in the target cell, and more innovation will push the limit in the future. I would like to highlight GenScript's enhanced lentivirus packaging system integrated with codon optimization, proprietary transfer vector, and our all-in-one packaging system. And lastly, also our comprehensive qPCR QCs. I would like to highlight our service with guaranteed functional title, upgraded packaging systems boost the functional generation. We can be as fast as 7 business days and competitive price with high successful rates and for large insertions. I'd like to end my talk here and listen to your questions. Thank you.
Thank you both so much for your informative presentations. We will now begin the Q&A portion of the webinar. So if you do have any questions that you'd like to ask, please do so now. Again, remember that Ask a Question box located at the bottom of your screen is where you can type that question directly into the text box. We will answer as many questions as we have time for. So we're going to go ahead and get started with our first question. This is for Roumen. Roumen, can GenScript design IRES for different applications?
Hi, yeah, we definitely can design IRES regions for different applications, including gene therapy and functional studies. We can also provide IRES options optimized for specific tissues or specific cell lines.
Perfect. Thank you so much. Our next question for you as well, Roumen. Can you develop targeted LNPs for my specific target?
Yes, so we do a lot of custom development for targeted LNPs. We can definitely work with you based on your specifications to develop the needed LNPs. And if you need any binding ligands, we can also work on those like protein, peptide, antibody. We can develop those ligands and we can incorporate them into the LNP. Thank you.
Fantastic. Thank you so much for that. Xiang, this one is for you. Can you share GLV3 vector and sequence with me?
Thank you. If you like to place an order of a lentivirus packaging service, our customized vector plasmid sequence can share with you. It's free. Remember, this vector provided here only for research use. Thank you.
Thank you, Xiang. And I believe this one is for you as well. Can GenScript's LVV be used in vivo?
Very good question. We have officially launched the purified lentivirus service in vitro, but we are running beta testing for animal use. Please contact with us and through emails listed here. Thank you.
Wonderful. And I think we have time for just a couple of more questions. So I'll get our last two asked. Or this one, I believe, is for you, Roumen. What quality grade are your off-the-shelf mRNAs?
Yeah, so the mRNAs that are off the shelf, those are all REO grade, research use only. However, if needed, we can customize them into preclinical grade upon request, or we can also work with our CDMO division if you need GMP grade to produce them for GMP procedures. Thank you.
All right, and our final question here for Xiang is, what is the purpose of providing control viruses to me?
Great. Oftentimes, our customers have different cell lines and also different protocols. They are not sure whether these reagents effective. We have a control virus with EGFP and cherry EF1 promoter that we already tested the efficacy, efficiency. So for various cell lines, it could be as a positive control for you and to verify the transduction process. Thank you.
All right, thank you so much. And with that, I do believe that is as much time as we have for questions today. So I'd like to again thank Roumen Bogoev and Dr. Xiang for your time today and your important research. Before we go, I'd like to thank our audience for joining us today. And I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Please visit GenScript's booth in the exhibit hall after the webinar for more information and resources. Questions submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand for 1 year, so until October 15th, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email out with your colleagues or anyone that may have missed today's live event. Until then, take care, everyone. Bye-bye.